Today, we're going to tackle one of the biggest questions out there. How in the world did life get started? And I mean, let's forget about dinosaurs or even bacteria for a second. We are going way, way back to a time before the very first cell, on a chaotic, primordial Earth, to explore how the simplest containers for life could have just assembled themselves from scratch. I mean, really think about that for a second. For life to get going, you have to keep all the important stuff, all those building blocks for RNA and proteins in one spot. But how do you pull that off on a planet that's basically just a giant, messy chemical soup? How did life build a room for itself before it even had the blueprints for the walls? Well, the answer might just be something called a protocell. Now, this is not a cell like we know it, with a fancy, complex membrane. Nope, it's way simpler. Picture a tiny, self-contained droplet, kind of like a bead of oil in water, that can form all on its own and trap other molecules inside. It's one of the top candidates for life's very first little laboratory. So yeah, the huge problem at the dawn of life was this: compartmentalization. You had to have containers. And the big question has always been, what did it take to build them? Did you need some rare, exotic ingredients? Well, some amazing new research suggests the answer is a fascinating no. Turns out the building blocks for these protocells were probably everywhere. They're called alpha-hydroxy acids, and scientists think they were formed by pretty much everything, from lightning strikes zapping the early atmosphere to being delivered fresh by meteorites. So the raw materials were just sitting there, ready and waiting. The real question is, how did they come together? So how do you get from a bunch of simple chemicals to an actual container? This is where the whole process gets really, really cool. To figure it out, scientists basically went into the lab to recreate the conditions of early Earth. They wanted to see if they could get these simple ingredients to build protocells all by themselves. And the process they found is just beautiful in its simplicity. You take these alpha-hydroxy acids, you heat them up to dry them out, which forces them to link up into these long chains called polyesters. Then you just add water. That's it. This simple little cycle mimics what would have happened constantly on a shoreline or in a puddle on the young Earth: evaporation under the sun, followed by rain. This schematic here lays it out perfectly. Over on the left, you've got your simple building blocks. Then in the middle, you can see how drying them out links them into these tangled up polyester chains. And then, here's the magic moment: rehydration. The second you add water, those chains just automatically pull themselves together into these distinct little droplets. And here is the incredible result. These aren't drawings. These are actual microscope images of the droplets that assembled themselves. They're tiny, spherical, self-contained worlds, all made from simple starting materials and a little bit of water. It's an amazing first step for life. But could they survive the next big test? See, life didn't start in some pure, distilled lab water. It most likely started in salty water, like the Earth's ancient oceans. So the next critical question was, what happens when you take these delicate little droplets and you dump them into a salty environment? Are they just going to fall apart? This is it. This is the moment of truth for our little protocells. They formed, which is great, but can they actually handle the harsh reality of their environment? Well, the data gives us the answer. So the first thing researchers noticed was that as they added more and more salt, the turbidity, which is basically just a way of counting how many separate droplets there are, it went down. A lot. Now, at first glance, you might think that's bad news, right? Like this salt is destroying them. But then they looked closer at what was actually happening to the droplets, and they found the other half of the story. While the number of droplets was going down, their average size was going up. They weren't dissolving, they were merging. They were growing. So why does this happen? Well, the chemistry is actually pretty straightforward. See, the droplets naturally have a slight negative charge on their surface, which makes them repel each other, kind of like tiny magnets. But the positive ions from the salt get attracted to that surface, and they neutralize the charge. Once that repulsion is gone, well, the droplets are free to just bump into each other and merge. This is a primitive form of growth, maybe even evolution, driven by nothing more than simple chemistry. Okay. So far, we have droplets that can form and grow in a salty ocean. That alone is a huge step. But the researchers discovered something else, something totally unexpected that makes these simple droplets seem uncannily alive. Yeah, it turns out these little protocells aren't just passive sponges soaking up whatever salt they happen to bump into. They have preferences. They're actually selective about what they let inside their tiny chemical worlds. The scientists measured the concentration of different salt ions inside the droplets, and what they found was just amazing. The droplets showed a really clear preference. They soaked up way more potassium ions, that's K+, than sodium or magnesium, even when all of them were floating around in the water. And here's the hard data. This is what scientists call the partition coefficient. Basically, a higher number just means the droplet is better at pulling that ion inside. And the winner is obvious. Potassium, K+, is at the top of the charts. The droplets are actively concentrating potassium while mostly keeping the others out. So why is this one specific preference such a big deal?
Here's why this is so mind-blowing. We think the ancient oceans were packed with sodium, but pretty low in potassium. Yet, if you look inside almost every single living cell on Earth today, from a tiny bacterium to the cells in your own body, you find the complete opposite. Cells spend a ton of energy actively pumping sodium out and pulling potassium in. And here we have these simple, non-living droplets getting to a similar internal state totally passively, just through basic chemistry. This could be a window into one of the oldest, most fundamental properties of life itself.
Okay, let's just step back for a second and look at the big picture. What does all of this tell us about the origin of life here on Earth? And maybe, just maybe, somewhere else. So let's just recap what we've seen. We know you can get these protocells forming spontaneously from common chemicals. We know that the salty ocean, far from being a problem, could have actually pushed them to grow. And, this is the big one, these simple droplets can create their own special internal environment that looks eerily like modern biology. It shows that complexity isn't some impossible hurdle, it's something that can just emerge from simple physics and chemistry. And as the researchers themselves put it, this is a pretty profound idea. A tiny chemical preference, a little more potassium here, a little less sodium there, that could have been enough to set one protocell on a totally different evolutionary path from its neighbor. This might be the very first spark of natural selection, happening not in a living thing, but in a simple chemical droplet. And that leaves us with one final absolutely tantalizing thought. The ingredients, these alpha hydroxy acids, they could have been delivered by meteors anywhere. The conditions? Wet, dry cycles and salty water. We think those exist or have existed on places like Mars or on the ocean moons Europa and Enceladus. If these unbelievably simple physical and chemical rules can give rise to such lifelike behavior here on Earth, could the very same process be unfolding right now, somewhere else in our own cosmic backyard?